I'd like to welcome you to the award presentation for the 2020 David G. Rognlie Award. This award is for meritorious discovery or advance in any area of structural science and can be awarded to someone at any stage of their career. It is intended to recognize an exceptional discovery or technical development of particularly high impact, rather than a lifetime achievement type of award. The award is created in the memory of Dave Rognlie. Dave was a dedicated and much-loved member of the crystallographic community, interacting over many years with a large number of scientists through his activities as owner of Blake Industries. Blake Industries sold diffraction equipment of many types, including Huber equipment. In 2014, he passed away, and funds have been donated to establish this triennial award from the ACA. The award is intended to embody Dave's values and personality, his generosity of spirit, optimism, selflessness, and unstinting desire to help others succeed in their endeavors. Dave played a particular role in the synchrotron community, supplying equipment to them almost from their inception, but had a broad and wide-ranging interests in science and the people doing science across many types of interests. It's awarded every third year, and winners receive a $3,000 honorarium and expenses to travel to this meeting. This is only the second time this award has been presented. The previous award winner was Helen Berman, who received the first award. For 2020, the award is going to James Holton. James is a beamline staff member at the Advanced Light Source, Lawrence Berkeley National Laboratories, and a faculty member at the University of California, San Francisco. James is being honored through this award to recognize his work in the development of beamline 8.3.1 at the ALS, one of the most productive beamlines in terms of both publications number and impact in the world. He is one of the most insightful proponents, designers, and visionaries in synchrotron beamline development. The innovations that James has designed, built, and installed have always been carried out with the goal of optimizing the process of beamline data collection. James made a number of pioneering contributions to the synchrotron experience. He's the sole author of Elves, which is the core structure solving program at the 8.3.1 beamline. He has pioneered the optimization of robotic data collection with developments that allow users to solve problems while still at the beamline rather than coming home with useless measurements. His colleagues and beamline users state that James is generous with his time and has done much to educate beamline users. Given the level of interest that Dave had in the ALS, not to mention the amount of equipment that he supplied, I'm sure Dave would be delighted to see this award go to James Holton. So James, congratulations, and I look forward to hearing your talk.
Well, thank you, Brian, for that very kind introduction. I'd also like to thank the ACA organizers for what must have been grueling reformatting of this meeting into the virtual presence that it is today. I really think it's turned out quite well. I'd also like to thank them for the honor of this award. It's really something. The thing I don't want to thank them for is making me follow Helen Berman. My God, that woman basically created the PDB as we know it. So it's a bit of a tough act to follow, but I'll see what I can do. I always like to begin with my most important slide, which is acknowledgements. I actually have 3 appointments. In addition to being an adjunct professor at UCSF and beamline scientist at LBL, I'm also a beamline scientist at SLAC in the SMB group. All these groups are supported by the NIH and Dewey BER, as well as the overarching support from BES for the light sources themselves. Now, the team for the work I want to tell you about today are Jane Tanamachi, Scott Klassen, George Meggs, who run the beamline, which we call the Tom Albertron. So it's named after Tom Albert, who was my thesis advisor, the guy who really built the place. And he's been a great supporter, friend, and advisor of mine throughout my career. I also want to point out Ward Smith, who was my program manager at the NIH for really my entire scientific career, from the Protein Structure Initiative through to last month when he very sadly passed away. Tom left us in 2014. I'm going to miss both of these men very much, but I hope to do them proud in the future. So the reason why I chose my trillion-dollar title was actually because of an effort to organize a meeting of my own, which would have been last week, the Diffraction Methods in Structural Biology, GRC. And I had a list of session titles of my own, but I also wanted to poll the user community and see what other people thought as well. I got some pretty good suggestions, but I was surprised to find that the number one problem faced in this field is money, which I thought was kind of disappointing, actually. I always thought the biggest challenges in science would be things like the phase problem that were intellectual or technological in nature. Because technology has two major goals. One is making the impossible merely expensive. Second is making the expensive cheaper or easier. And although it can be difficult to do both of these things at the same time, I think that's kind of fun. So what is $1 trillion? Well, it's not infinity, okay? It's Apple, that amount of resources. And I calculated $1 trillion because it's my estimate of the value of a cure for cancer and heart disease. So that's just the average salary of a human being times the number of people who die of these diseases every year, times adding 3 years to their lives. That's the value added to the human race, if you could do that. But what we're used to thinking about is the amount of money you normally get as a single PI on an R01 grant. And you can do a lot of really great things on an R01 grant, but it's largely limited because the NIH itself is limited. Their total budget is only about $40 billion. And if you search for all the grants that even mention the word cancer, it's about $6 billion. A good fraction of that, but it's not $1 trillion. So something that's $500 billion would have been, say, the amount of global spending on fixing the Y2K bug. That wasn't even a real problem. But a better example maybe would be the NASA Apollo program, which was, in adjusted dollars, $300 billion to go to the moon. So for $1 trillion, we could not only go to the moon 3 times, but repeat all the technology development that had to happen to make that possible. That is what $1 trillion is. And just because it's a question that usually comes up, the Department of Defense is not quite $1 trillion. And it's very difficult to talk about this amount of resources without resorting to politics, but I think that's the wrong way to do it. We're scientists, we're not politicians. If you think you're a better politician than a scientist, then by all means, run for office. But as a scientist, technology and science are not about taking something away from one thing and giving it to another. They're about creating something new, about saving the lives of people who would otherwise not continue to live. That's value. That adds value. And if we could come up with a plan that would work, and we could explain how it works, then the resources will be available. Total amount of money available on Earth is about 80 trillion. That's the sum of all goods and services of all the people on the planet. So if for $1 trillion, we could save the lives of more than half of the people on Earth who are going to die of these diseases eventually, then that's quite a bargain. The US federal budget for this country, that's only about 5 trillion. But the amount of money that the US spends on healthcare, not just the government, everybody, is a little less than that. And a good point of reference is the CARES and HEROES Acts for COVID-19, which were just passed. Again, $1 trillion to keep this kind of damage to our civilization from happening is also a bargain. By comparison, the amount of money spent on, the amount of NIH grants spent mentioning SARS last year adds up to only about 12 million. Viruses in general, about 1.5. But that's a pretty substantial fraction of what the NIH does, considering all the things that the NIH is responsible for. So what if the NIH had $1 trillion? Well, that would be enough to fund all the applications that were sent in for the last 5 years. Not just the ones that were funded, not the ones with good scores, all of them. How many of you have a grant that got triaged? Was it because it was a bad idea? Another thing you could do with that is buy 100,000 Titan cryoses and give them away to everyone who needs one. And you'd still have enough left over to build 1,000 X-Fells. Or another way to maybe be more equitable about it is there's only about 100,000 biological scientists in the United States. So if you just gave each of them 10 million, that would add up to $1 trillion. Now, I'm not just talking about the PIs, the postdocs, the students, even the technicians, 10 million each. What would you do with that? Would that solve all of your problems? Well, you probably wouldn't fly coach anymore. And it'd be nice to be able to get all the resources you need. But in order for this to work, it has to work. It has to solve all of the things standing in the way between what you got into science for in the first place and actually achieving that. Those are the intellectual and technological challenges that I believe are the real purpose of science. So getting back to my critical challenges, let's take that first one. Predicting crystallization outcomes. Could we actually do that with $1 trillion? Well, the biggest problem with predicting crystallization outcomes is actually the data aren't very good. The PDB does contain crystallization conditions, but they're all one-offs. They are full of errors, etc. And it's better than nothing, but what if we just did it all again? Reclone, re-express, re-purify, recrystallize, resolve everything in the protein data bank, including the membrane proteins. Now, I've talked to a few people who know what they're doing with this, and we think it would add up to, generously, about 500 million to redo the whole PDB. And since this is data, and data is better with error bars, let's just do that 10 times. So $5 billion to collect the data. Now we have to analyze the data. Stands to reason that we're going to have to do some structure prediction to do this. So, well, Google did pretty good with CASP last year, so why don't we just buy them and tell their machine learning people to solve this problem for us. Put that whole advertising, machine learning stuff on the back burner for a while and work on this problem. And Facebook's doing this too, so we could buy them as well. That way we got two different teams working on the same problem for us. That still leaves enough money left over to buy some computers. About 10 zettaflops at current pricing rates. That should probably get a lot of work done with those. The question, though, is would it actually work? I think it probably would. That might even be overkill. But the bigger question is what would the impact be on your research? What if you could just type a sequence into Google and get not just the crystallization conditions, but whether or not that protein will crystallize? How much of an impact would that have on structural biology, on medicine? Well, and by the way, that computer is good for other things. You could actually use it to solve the fates problem by brute force. I'm not really kidding about that. 10 to the 21 flops. Let's say we give 5 CPU minutes to each guess of the phases and just run auto build on that. We got 1 year of runtime, let's say 2 phase values per HKL. That's enough to do 10 to the 27 combinations, or 89 HKLs. And for a modest cell in a reasonable space group, that's everything out to 10 angstroms. That's respectable. That might be enough to crack it. Because you don't have to do everything out to 10 angstroms. You could just do the brightest ones. They matter more. There's various other optimizations you could do. But this amount of computing power, not even $1 trillion, might be enough to actually crack the phase problem. But again, the question is, if it works, how many of your problems would this solve? How often are you stuck not being able to solve phases? But I think the ultimate goal of all structural methods is something I like to call the magic microscope. And that is, imagine you had a device that would let you look at a single molecule at any resolution in space or in time. And you can continue watching that molecule for as long as your heart desires. It won't damage it, or the patient that it's sitting in. You have the ability to tap on it, tug on it, mess with it. This device would be handheld, widely available, millions of them. If you had such a device, my question to all of the biologists I know is, what would you look at first? The answer is easy. It's whatever you're currently working on. So a better question is perhaps, what would you look at second after you wrapped up that other project? And it wouldn't take you very long to do it. But if such devices existed, what would you look at? Would it be a large complex? Would it be an enzyme active site? Would it be something you had never even thought of looking at before because you thought it was impossible? But also, if we had such a device, how long would it be before the end of disease as we know it? I don't think it'd be less than a year, but I don't think it'd be more than 10 either. So I've actually been thinking about this problem for a while. I made this picture back when I was an undergraduate. I had this idea of stretching proteins. I had recently read the paper by Carlos Bustamante where he had been stretching DNA molecules between two beads and measured their elasticity. It was one of the first true single molecule experiments. And I wanted to try it on proteins. I figured if I could pull the heme out of myoglobin, that might be spectroscopically visible. And I worked on this for about 3 months until my advisor at the time, well, in his words, said, why don't you work on something that has a better chance of working? But I still stuck with it. And it was a project I wanted to do when I went to graduate school. And when I was interviewing at graduate schools, I met a man named Tom Albert. And what he told me about my idea was, let me tell you why it's not going to work. He told me about this big protein called Titin, which was a micron long, 37,000 amino acids, the largest known single chain of any protein. And it runs from the M line to the Z line in the sarcomeres of most muscles. And it stretches as part of its function. And what he said was that the individual domains of this protein actually denature cooperatively, so he wouldn't learn anything new about folding by doing that. But like most students, I listened to the part of that I wanted to listen to, and I realized, wow, Titin is a micron long. That means Carlos Bustamante can stick this in between his beads. He can use the system he's already got. I might tell him about that. I did see Carlos maybe about a month later, and we called Tom and found the paper and showed it to him, and he was very excited about it. I thought it was exciting too. But through the fullness of time, I ended up not going to Oregon. I ended up working for Tom at Berkeley. And a few years later, this paper came out. Not just Carlos's paper, but it was back-to-back with another science paper about exactly the same experiment, which was sort of back-to-back with a third paper in Nature. So not just one science paper, but two, and a Nature paper. A lot of my friends and classmates thought I would be upset about this. I wasn't named on any of these papers. It was fine. I didn't do any of the work. But I thought this was awesome because it proved that I wasn't an idiot. So what I learned from that experience was just because it's a crazy idea doesn't mean it's impossible. So when I got to graduate school, I started learning about this X-ray crystallography thing, because when I was an undergraduate, I was doing NMR. And what I found I was doing was I was just really transforming data from one format to another using programs that I was driving with scripts, which is something I still like to do. But you feed a script into a program and it generates new text files for logs and data. And you take those numbers and put them back into the script or into a different script. And I realized what I was doing was I was just moving numbers from one part of the computer into another. I thought, well, why can't I write a smarter script that would do that for me and maybe allow me to just connect all these scripts together into one big long chain that would go from diffraction images to PDB files. And this became something called Elves, and that was my doctoral thesis, actually. And when Tom hooded me for my PhD ceremony, he said that the only contribution he made to my thesis was telling me it was impossible. Man was nothing if not understated. And he's pulled that trick on a few other students since. But Elves really did work. It was, I believe, one of the first programs that could just take some data and some English commands, run some standard popular programs, and give you a structure out in the end. And what I learned from that experience is it turns out the algorithms that have been developed by our ancestors actually work, provided you don't make any mistakes. And you make mistakes when things are too complicated. So while I was writing my thesis, Tom started building this beamline up at the Advanced Light Source at 831. And I thought, you know, that might be a good place for me to test out my Elves, because we're better than the very place where the data comes from. And I asked him if he found anyone to run it yet. And he said to me, I don't know. Do you know anyone? Couldn't actually bring himself to ask me to apply for the job. I don't know. I guess some people thought it would be career suicide working at a beamline instead of following the usual route. But I thought it'd be kind of fun. I was young and naive back then. One thing I did notice is that everybody had different pins, and I started collecting them. These are just a few that I've picked up over the years. And I figured, you know, we need to have a way of handling all these problems. So I made a goniometer and a set of tongs that were just sort of a superset of all the different sorts of pins that we came across, including the old Yale pins, which are the IUCR standard. And the super tongs just epoxied them under the end of some pencils. And those may look like number 2 pencils, but they're not. They're number 3 pencils. We use the conductive lead in the middle to heat them so they don't ice up. And a lot of you have seen these foam dewars nowadays. These actually had their genesis at ALS when John Spear was working back then. I like to modify them, but we'll dent in the bottom for pucks, and I also add some magnets on the side, so you always have another crystal ready to go whenever you go in the hutch. Also, when they ice up, a wash bottle full of liquid nitrogen is not very safe, whereas this is a commercial surgical tool that we found works pretty well. Another big problem, which is still with us to this day, is people not knowing where the beam center is. And so I decided to solve that by making a semi-transparent beam stop. And it turns out lutetium is the right thing to make it out of, because it has the flattest absorption curve over the X-ray range of interest. And so anytime you can't find your beam center, just look at the image. There'll be a little black dot right where the beam's supposed to go. Also, back in the day when data collections took hours, people, or if the beam came up and down, people wanted to know, and they may not have had cell phones. And so we set up these little pagers, and if you just took one out of the tray, it would alert you whenever something interesting happened. Also, everyone comes with different kinds of hard drives, and rather than try and have a single solution, we just made a little octopus of cables, so you plug in whatever format you've got, and then everything automatically gets backed up. I also noticed that people kept touching the computer monitor, which puts fingerprints all over it, but I figured, well, if they're going to do it, it might as well do something useful. So I laid a touchscreen overlay on top of the monitor, and now whenever you touch a part of the screen, it moves that into the beam. Much more intuitive interface. We also found that people didn't always know when the beam was off, usually because they didn't close the shutter properly. So I actually bought this traffic light on McMaster car. That's an actual real traffic light. It's hooked up to an ion chamber in the hutch. And anytime there's no x-rays, it turns red. Anytime there are, it's green. But I still have people occasionally tell me that something's wrong and there was no beam. And I ask them, is the traffic light red or green? And they say, what traffic light? And I do spend a lot of time studying my users. I hope they don't mind that too much. But I'd like to make sure things are nice and efficient. For example, of the total amount of time we spend collecting data is about 50%, but the rest of the time breaks down in a rather interesting way. Mounting and centering, which we focus on so much as bottlenecks in beamlines, are actually, even back then, when we were mounting by hand, a relatively small part compared to the time between the end of a snapshot and when you do something else, and the end of a data collection when you do something else. These are the time when the user is thinking about something, or preparing, or maybe just not prepared at all. And so by reminding people when the data collection is about to finish, and making strategy steps faster, we can reduce these overheads. So what I learned from watching users is that when it comes to methods development, nothing beats sitting down right next to somebody and paying attention to the things that are bothering them as they come up. I suppose these days we can all appreciate that one. We also decided to try and eliminate crystal mounting altogether using an in situ mounting idea. This is one of our first in situ goniometers. We're still using this to this day. And here it is with our newer detector. What I particularly like about this one is it's still zero parallax optics. So the microscope looks down the X-ray beam, even though it's not directed down the X-ray beam. If you have a mirror at the right angle, you can actually look straight down the beam, even if it's only half the numerical aperture. It took me a while to convince myself of that, but it really does work. And the microscope rotates with the sample so that even as it rotates, you can still see that your crystal's in the beam all the way around. But what I learned from in situ is trays dry out when they're too thin, and when they're too thick, they have too high a background. So what we really need someday is some of this stuff. Now, of course, I was interested in just how often the Elves could solve data sets and see how well they worked. I was surprised to learn the answer is almost never, but I'm not alone, because if you think about it, the average data set in the world takes around 100 seconds to collect. There are about 200 operating days a year of most synchrotrons and about 150 beamlines of one type or another around the world. This is the capacity to collect 26 million data sets per year, but the PDB only got about 13,000 data sets published last year. So there's a gap. And I've spent a good amount of time trying to figure out the reason for that gap. Unfortunately, I backed up all the data. I used to do these by hand, and then we bought this robot for writing DVDs one at a time. It triggers when there's enough data to merit one. And I've now gotten to the point where I can start restoring all that data onto spinning disk. This is a machine I call Petabyte, which has 90 drive bays. And if I fill that up, once I do fill it up with 12s and 13s, it should get up to a petabyte once it's full. And by looking at old data, you want to look at very old data, because data that was just collected, if you can't solve it, you don't know why. Whereas something collected 15, 20 years ago, if it's not in the PDB by now, it's probably not going to be. And so looking at just the MAD data sets from that early period at 831, I found that about half the time was spent collecting data, and only about 1 out of 5 data sets are easily processed. That is, they give you iris sigmas greater than about 10. But if you look at how many of those were published, about half of them are in the PDB. Another 1 out of 5 or so, again, were published, and it was just a different form that we had at the beamline. But a lot of them were unknown, or but also unique. So showing this graphically, if you try doing a molecular replacement rigid body refinement on the closest things in the PDB, you find that for some things, the R factor is nice and low, and the R ISO, the R factor between the data in the PDB and the data on disk, are also low. Those are structures that were actually solved and published. These ones that refine well but have very different data are non-isomorphous forms of the same protein. These guys, however, are completely unique unit cells that were just never solved. So I've taken a look at some of those. Of those, about 1 out of 5 are twinned. And if we look at the unpublished forms, again, twinning is part of the problem, but pseudotranslation is also a really big problem. This is looking at solutions that we were able to solve using sequence-independent molecular replacement. So, but if you look at the PDB itself and just search for the word twin, you only find 0.14% of the entries. So twinning is definitely underrepresented in the PDB, and apparently a much bigger barrier to solving structures than we previously realized. So what I learned from looking at all that old data, at least so far, the sample is indeed everything, but there are a lot of places where methods can improve. And to address that, I actually started simulating data, because in all those 200 terabytes of data that I've got lying on disk, nowhere is there a controlled pair. So what I'm doing here is showing you the result from Molfsum. This is because Molfsum is a major processing package. Molfsum is processing in reverse. And point of programs like Molfsum is it makes data on the photon scale, the actual scale of the detector. And that means that everything, like particularly the photon noise, is on the right scale. When you process these simulated data, you get statistics that are very, very close to those of the real data. And the noise level is similar. But one of the most interesting things I found with Molfsum was that if I simulate some data and process it with Elves, I get R mergers that are perfectly normal, but the R work and the R free are way too low. They're down in the single molecule or small molecule levels. But if I simulate a multi-conformer model, which is harder for automated building to construct, then I get realistic R work and R free. So that implies maybe something's wrong with Elves. But if I short circuit it and take that starting model, the right answer, and drop it into refinement, I then, again, get these same R work and R free. So what that tells me is that the data aren't the problem. The data actually faithfully reflect the real structure. We just don't have the technology to properly build what the electron density is telling us. So that I thought was a really important and a big revelation for me, was that the R factor gap, the big gap between R work, R free, and R merge, or R mez, is a modeling problem, not a data problem. And the other really important thing I learned from Molfsum is that 1 + 1 is 1.4. I know that's said a lot of things, and that may not make a lot of sense, but it really is true that if you're adding noise from different sources together, they add in what's called quadrature. And square root of 2 is about 1.4. The implication of that is that in any given experiment, there's one and only one source of noise that's ever really important, and changing or reducing any of the others won't make any difference. Now, when you become a beamline scientist, you very quickly realize you need to learn a lot more about radiation damage. And that's because RadDam limits pretty much everything that we do when it comes to data collection. This is what happens when you thaw a sample that's been previously shot. But the most fascinating and shocking things that I learned early on was that radiation damage is indirect. So this is an experiment where I was measuring how fast selenothionine gets broken by the X-ray beam. And what's surprising about that is the point at which half of the selenothionine is already gone is well before the time when you know from physics that half of the selenium atoms have been hit by X-rays. So the energy is actually being absorbed somewhere else in the sample and then transferring through the bulk to a selenothionine and then breaking it. So it's actually doing chemistry at a distance in the solid state, which is not the thing that I expected back in the beginning, but I think it's fairly well established now this is how this works. But that was a revelation for me. And of course, if something's that indirect, it's intoxicating, there might be a way to actually interfere with it. So I did a lot of little experiments with selenothionine, trying to find additives or conditions that made it decay faster or slower. And I found a lot of things, and if you add them all together, you can get some big effects, almost a factor of 8. But problem is, most of these things we don't have a terrible amount of control over, and the individual ones are never really all that much bigger than a factor of 2. And the problem with that is, okay, a factor of 2 may sound like a lot, but it's only, because 1 plus 1 is 1.4, it's only a 40% change in signal to noise. It doesn't really make or break solving math structures as much as we'd like. So what I started doing was just start writing down world records. And if you go through the literature, there's unfortunately a rather short list of all the different kinds of chemistries that have been calibrated, as in how many megagrays does it take for that reaction to go halfway. And I posted this up on the wall at the beamline because these are not goals to be achieved, but limits to be avoided. You don't want to set new records when you're collecting a new and difficult data set. A lot of people have asked me from that chart what's a megagray, so I made a little table where I went around through the biosync database, and based on the flux and beam size of all the world's beamlines, it'll tell you how many megagrays per second any given beamline is doing. And I've made a lot of friends that way, but it's also, I think, a handy tool. I've since converted that into a web page where you can actually pull down which beamline you're at, or just enter in the flux. And this particular one here actually will predict whether your experiment will succeed or fail. So you put in your goal, the experiment, and down at the bottom, it'll tell you the number of crystals that you'll need to succeed based on limitations known from the literature and from first principles. You want this number to be less than 1. If you need more than 1 crystal, it doesn't mean it's impossible. It just means you might need to merge some data together. All this is based on this big incomprehensible equation, which I'm very proud of. I won't go through it today, other than to point out that flux is not part of this equation. Neither is crystal symmetry, nor the exposure time. And that's because radiation damage is self-limiting. The math actually works out this way. It's dose comes from photons per square micron, so does scattering. So the number of photons that you're going to get out of your sample before it dies is actually a fixed number for any given sample. It doesn't matter which beamline you're at. It's actually around 1 million photons per cubic micron of crystal under cryogenic conditions. And the only thing that seems to be able to break that is the exfil. You get about a factor of 100 more, or by going to room temperature, when you get a factor of 100 or so less. And I've been making this graph of really just that, the dose rate dependence of damage. The dose rate effect is one of those really elusive things in the radiation damage field. Everyone would like to find it. Everyone's found one, maybe a little bit there. And I try to put it all down on this graph for perspective. So on the x-axis is the dose rate in kilograys per second, and the y-axis is the maximum tolerable dose. This is a log-log plot. So pretty much everything we've done at cryo is around the same, around 30 megagray. Room temperature is between 50 and a few hundred kilograys. There's one shot, this is Mario Schmidt out here at the biocars, but clearly a very different world at the X-Felt. There's a very large and unexplored territory here in between. I wouldn't want to be the one to try and draw a straight line through this. We need to fill it up with more data points. But the long and the short of everything I've learned about radiation damage is it's kind of just unavoidable. Even X-Fells have a certain type of damage that happens under those high fluences. So you have to deal with it, control it, mitigate it. But what I love about radiation damage is the community. It's amazing how many people want to know about it, even though it's not supposed to be an interesting subject. And the meetings and research that's done is really amazingly collaborative, because it's great when everyone gets together just to try and figure out how to make something work. Now, XFELs are an interesting story as well, because one of the problems with XFELs is the data quality is never as good as we'd want it to be. And so I've been turning my simulation energies towards trying to fit not just not a million images, but just one. Can I actually recapitulate a given image? This is from a paper out of Axel Brunger's lab a while ago. And if I tweak every parameter I know of, I can get this image. So that's the real one, and this is the simulation. You can see it's under predicting. But if I increase the mosaic spread, it actually makes things worse. It predicts more than it should. So what I did is I said, okay, let's just tilt the crystal around all three axes and calculate the correlation coefficient between the two images, the real and the fake. And you can just plot those numbers on a 3D grid. A 3D grid is an electron density map, and you can look at that map in COOT. So what you're seeing here is called a texture. I actually learned that word here at the ACA meeting. A texture is the three-dimensional histogram of the orientations in the crystal. So these two little sausages are actually not so much two crystals, but two plates. So these crystals are known to grow as plates. And those two plates are at slightly different orientations. So there's two blobs, and they're stretched because those plates are bent. But reciprocal lattice points, which look like this, are not round. They have structure. And by ignoring that structure, it actually introduces significant errors in the data. So I can do a 3-blob simulation, which models after that, those blue sausages, and I can fit much better to the real image. See, that's the 1 blob. That's the 3 blob. It's not perfect, but it's better. And then I can ask, okay, is it sufficient but not necessary? And if I look at the statistics from data that was simulated with 1 cluster versus 3, it works a lot better when there's only 1 mosaic blob versus a more complex structure. And in fact, I think this almost completely explains the larger R factors we see from processing stills data. This doesn't mean stills are bad. Stills are actually theoretically the best way to collect data because they have the highest signal to background ratio. There's no background accumulation with rotation, et cetera. But I think the way forward with data processing is that texture and modeling texture is what we really need to do next. Now, simulations have also been useful for debugging software in other ways. This is a simulation of a selenium-methionine data set, which is absolutely trivial to solve. And this one is completely impossible. No one I know has ever been able to solve this without cheating. Can you see the difference? Yeah, don't worry, this screen hasn't frozen. The change from selenium to sulfur in a protein is actually really hard to see by eye. But if you interpolate between those two images, you can then get a linear scale of increasing difficulty. So fraction sulfur, and you can plot on the y-axis correlation coefficient to the right answer. We know what the right answer is because it's a simulation, and difficulties on the x-axis. And you can run different programs, and some will do better than others. And whenever one does better than another, the author who didn't win is usually interested in why. And looking at the data just before the point where it falls over is extremely informative. And what happened in this case is everyone started playing this little game of leapfrog, where Phoenix Autobuild did pretty good, Crank 2 did better than that, ARC did better than that. Current champion is actually Tom Terwilliger, who was able to solve a structure manually this way. But the really cool part about this is by using the changes that Tom made in Phoenix, we were able to solve this structure. This was intractable by every means we tried before those changes, but by pushing the software in a direction towards weaker signals, we actually took unsolvable data and made it solvable for one of my beamline users. So for me, that's pretty gratifying. So what I've learned from doing these challenges is that community efforts really make a big difference in technology development. The methods developers really need to work with data that's right on the edge in order for them to find the key ways to increase the power of their programs. Now I want to tell you about COVID. This is a beamline scientist way of looking at 2020, because we were collecting data merrily away right up until California lockdown here in mid-March. That little jump there is some of my users from UCSF. And when they came back, they had been growing a lot of COVID-19 crystals, and that's the first diffraction pattern they got. I thought they were shooting lysozyme or something, but that's actually macrodomain. And so this consortium is now called QCRG out of UCSF, and they are sharing all these data openly. This is was a release done just there. And it's really been an amazing leap forward. Getting things going again is gratifying, and also screening for possible ways to do something about COVID. And I want to tell you a little bit more about a side project of mine, because when all this started, I was sitting at home actually sick. I was coughing, had a high fever, and I couldn't get a test. So I started thinking about how I could do it myself, in my kitchen. And I figured what the world really needs to end the pandemic in a 48-hour day is to test everyone on the same day, and they all get the results at the same time. Now, the problem is if tests are too expensive, no nation in the world can afford to do that. So we need them to be cheaper. PPE is in short supply, especially in the beginning of the pandemic. But so what that means is that if you test yourself at home, then there's no danger from yourself, so you don't need the PPE. And a big unappreciated problem with testing is logistics. The fact that you have centralized testing centers means you need to do shipping. Amazon ships about 1 billion packages a year. So doing 7 billion in a day is just absolutely intractable. So we need a decentralized system. Also specialized equipment. I don't actually happen to have a PCR machine in my kitchen, nor do I have a centrifuge, but all the basic things you would normally find around a lab, hot water baths, for example, are also difficult to do. So what that means is we need to take advantage of any hardware that's already out there in everyone's home. Specialized skills are rare, so we need tests that are easy to do, preferably one where anyone could do it just by watching a YouTube video, like a cooking show. And particularly early on, antibodies might be reliable, might not be. Enzymes perhaps may work the way we want them to or not. And so reagents that are both robust and pandemic compatible, hand sanitizer and toilet paper, for example, are not things you'd want to have to use. So here's my idea. You ship the reagents in blister packs, which are readily available in large quantities as dry chemicals, and then you have the end user simply spit in it. Mix that up, maybe boil it in a hot cup of coffee. Most places on Earth have the ability to heat water. And then absorb the RNA out of the solution preferably onto a surface, and then put that directly over the camera of a smartphone. The blister pack will act as a reflecting mirror to get all the light into the camera. Now, the flash LED will have to have a filter on it, but the idea is you use a blue excited fluorescent dye that is in this is called a molecular beacon, which is a self-quenching hairpin that has a small compatible sequence with itself, and the rest of it's compatible with the virus genome. So if there is virus around, the beacon will open up, exposing the fluorescence, and the fluorescence increases by about a factor of 50. And that light in the camera would then indicate that you've got virus. So why aren't we doing this already? Well, because the level of fluorescence when you haven't amplified anything is awfully low. But I was surprised to find that we might be able to detect it. Smartphones, believe it or not, have already been shown to detect only 1 million particles per mil. That's 10 to the minus 15 molar. This is a paper in Nature a little while ago. And guess how many viruses are in 1 mil of saliva around that many, according to a recent article in Lancet. Now we can boost that signal, perhaps even by a factor of 1,000, by just including more than one tag, just anneal to different parts of this really extremely large genome. This is the biggest RNA genome yet discovered. Also, I think working under denaturing conditions means that there won't be any RNase around if you're working in, say, 5 molar urea. You won't have any enzymes either, but if all you're doing is molecular beacons, you don't need enzymes. So my plan A is to try out this molecular beacon idea, see what the detection limit really would be. And if that doesn't work, try a time resolved approach, which I'll show you in a second. So the sample holder might look something like this. You'll filter over the LED, another filter over the lens, and you need to have some kind of color filter on each side. But aluminum foil bent around an object is actually remarkably light tight. I do this for all of my photodiodes. And the CMOS sensor in a smartphone can be thought of as just a really fancy photodiode. The sample itself would be a molecular beacon this way, and this is my little drawing of the genome holding all this together. You could also use some of these annealing sites to attract the genomes to the surface of the glass. And because there's no lens, you're maximizing the solid angle of light that will go into the CMOS sensor. And there are papers, in fact, written about the spectral characteristics of these sensors. They're really quite remarkable things. I remember back when I was starting out as a beamline scientist, a back-thinned illuminated device was a dream that was really expensive and hard to get. But now, thanks to the demands of cell phones, we've all got one in our pocket. And these, the way these things distinguish colors is they have little color filters that are laid out on top of the sensor itself. And so you want to read this thing out in RAW mode in order to get rid of all the JPEG corrections that would get rid of small signals. But this is basically taking advantage of an old trick in X-ray crystallography, is that if you want to get much higher detected quantum efficiency, all you need to do is throw away your point spread function. So this is the spectrum that comes out of the LED on your average person's phone. And these things are incredibly bright. It's about 4 times 10 to the 17 photons per second. That's impressive. And so there's actually quite a lot of light to throw away, potentially. So you can't use the whole thing because there's, you need to differentiate colors. But if you search around with different glass filters, I found one that if you multiply those two curves together, you would change the emission spectrum of the phone to be a pretty narrow blue band, which matches really well to that of ADO 465. This is one of the commonly available green fluorescent dyes. Now, the emission of ADO 465 is a pretty high Stokes shift, so that makes this a little bit easier. But it's not completely clear of the blue lines. So this is the light that's bouncing around in the sample, except that the blue light is probably going to be about 1 million times higher than the emission from such a small amount of fluorescent dye. So we have to knock that down somehow. It is already very compatible with the green receptor in the CMOS sensor, but by applying yet another glass filter, we can squash that down, shifting it a little bit, but giving us a nice black zone in between the two colors. So these would be the two different filters that I'm thinking of trying next. They're actually in the mail. Now, if that doesn't work, we can take advantage of something quite remarkable, that the CMOS sensors have what's called rolling shutter. A lot of modern X-ray detectors do this as well, and you probably noticed that if you ever try to take a photo of a fan, because a spinning fan will have these weird structures to it because the sensor is not reading out all the pixels at the same time. It actually starts at one end of the detector and works its way over to the next. So I actually decided to try and measure what sort of time resolution we could get out of this. This is a photo I took with my phone in the dark in my bathroom, using my wife's phone to take a flash that's out of sync with it. Maybe you think it's easy explaining to your wife what you're doing in the dark in the bathroom with her phone. Well, Christine's a scientist, so she understands. And it turns out you can get time resolution of a few hundred microseconds fairly easily, just with that crude little experiment I just did. And there are fluorophores that last longer than that. Most of them are lanthanide based, but so you're not going to be able to do anything with ADO, but with terbium and europium salts, this has been demonstrated to be long. So what happens here is that you flash the light, and then as soon as it's dark, there's nothing left except for this fluorophore glowing all by itself, with all the other light will have faded away. So things I could use some help with, if you know anyone, is designing 1,000 oligos for the SARS-CoV-2 genome. Concentration steps that could be easily done by a novice are tricky. I haven't found a lot of good literature on hybridization in urea. I'd like to know more about that. Anyone who wants to try a filter and a dye and test it on their phone, this is pretty easy to do. I'm literally in my garage right now doing these sorts of tests. And establishing what the detection limits are will make or break this type of approach. But more than anything, I'd like you all to tell me why this is impossible, so we can figure out together how to make something like this work. So what I learned from all this is crazy ideas are possible. I did learn that a long time ago. Simplifying things makes them work better, more efficiently. You got to watch the user, see the kinds of mistakes that they make, the things that get in their way. The sample is everything. 1 + 1 is 1.4. And if we can keep the basal fluorescence below the noise floor of these sensors, then it won't be important. But most important of all, community improvement of technology is the best way to go. And thank you all for listening. I'll take any of your questions.
Okay, James. There was one question in the Q&A, which you may have answered already, but on using a salad spinner as a centrifuge.
Yeah, the salad spinner, well, we have one. It gives you a few Gs, but it's actually not quite as good as just spinning a rope around your head. And I've done that one as well, but it's really tiring. It's difficult to get more than about 10 or so Gs with anything you got in your house. So it might be enough to settle some yeast, but do an actual 1,000 G spin like we're used to doing in the lab is not really accessible at home.
What's the major technical challenge you think on pushing, I assume, the R values of protein structures down to a similar level as small molecules?
Okay, so that's something I'm actually is part of my current research. And what I've found is the residual R factor is divided almost equally between the protein region and the solvent region. So you can actually get significant gains of up to about 5% in R factor by improving the way you do bulk solvent. The best way we've found is to use an MD simulation, which isn't as expensive as you might think. It takes like 8 hours or so to get a much better bulk solvent mask. And then what you see are difference features in the protein. And the point where I'm hung up right now is that proteins are multi-conformer things, even at low resolution. And the solvent is sensitive to that. And so unless you've got the right combination of conformers, like A versus B, B versus A, then things still get hung up. And so that's what's limiting now. But it seems like if you get the conformations right, the constellation of multi-conformer stuff correct, and do a simulation of that, you should be able to get pretty low. I've gotten some R factors down into the 8 or 9 level. Sandy
Eagle has a question, well, it's actually more of a point, saying that there's a YouTube video on how to scramble an egg in the shell. The method might work for the centrifuge idea. But from Ilya, saying, great talk. I believe that for solvent-dependent crystal to become usable, it must lose about 10% of its solvent. What radiation damage does, is it necessary to destroy the whole crystal or just a portion of it? Meaning the dose should be computed for, let's say, 10% of the crystal volume.
Okay, I'm not quite sure I follow that. I do know that at room temperature, a significant amount of the damage actually is due to desiccation, because you convert water into gas by reaction with the organics. And it's about 18% of it that gets turned into gas at the room temperature.
Stephen Kelly, who says, you mentioned twinning as a major problem in structure solution. Do you think that most unsolved twin data sets that have been collected at your beamline could be solved with enough time devoted to processing? Or are there fundamental problems with the data that would make deconvolution effectively impossible for most samples?
I would say, yeah, I think that they should be solvable. In fact, I know Peter Zwart is actively working on the problem of solving anomalous structures in the context of twinning. But it is definitely a major gap in the software that we currently have. Twinning is just not something that we know how to do with anomalous. So you can solve twin structures with molecular replacement reasonably well if you are lucky enough to have only one copy in the asymmetric unit. And there have been a few twin structures solved by anomalous, but not many. But it's something that a lot of the authors I know, George Sheldrick and Peter as well, are interested in doing this, but they've had a hard time justifying their work because there's so few of these in the PDB. So my hope is by getting this information out there about just how many unsolved structures there are that are twinned and with pseudotranslation, that they'll have a reason to do that work.
Okay, next question from John Hilywell. Hello, James. Congratulations on a great talk. Thank you. As you know, there are various synchrotron updates going on, e.g. ESRF, EBS, Diamond 2 in the pipeline. Is there also an ALS2 new lattice project? I'm sure your management will be very happy when you answer that one.
Oh, yes, there is. The ALSU, they're calling it, is scheduled for around 2024. And that will make us go dark for a year. And the improvement to my beamline won't be really noticeable. And so I'm not too happy about that. But there definitely are ways to improve the optics in order to take advantage of the upgrade. The beam will be essentially diffraction limited in the soft X-ray, but that's 4 years away.
Right. It is in the funding pipeline.
Yes, it is. It's CD3 now.
Right. Okay, so Eddie Snell, EMBL, we're playing with the CD-type sample holders for SACs. Could provide a container with an ability to spin. I don't know how many Gs, or am I a Luddite still having a CD player? Okay, so let's see. Ilias, I'm going to see if we can do better with this. Great talk. I believe that for a solvent-dependent crystal to become unusable, it must lose about 10% of the solvent. With radiation damage dose, is it necessary to destroy the whole crystal or just a portion of it, meaning the dose should be computed for 1/10 of the crystal volume? And I think that may be related to some of the small beam measurements where people are, taking a beam across the crystal and using it up slowly, burning it up slowly, but that's my thought.
Okay, well, dose, so the photoelectrons travel a few microns. And so the dose to the crystal isn't really a unit cell by unit cell thing or a solvent versus protein thing. The electrons go everywhere. And so the energy is more or less evenly deposited on the micron scale and smaller. On larger scales, yes, you can have small beams where a lot of the energy can escape the illuminated region and does damage to things around it. And that's an advantage when the crystal itself is really small. But I think the best thing to do with really small beams is actually to blow them up by moving them, by rastering them, because that gives you information about where in the sample a photon came from, and that will allow us to essentially paint with the X-ray beam and then decide after data collection which parts of the sample we wanted to get our data from. Like if the crystal's non-isomorphous in one half versus the other, then
so you're rastering but as you're moving the beam, you're you're recording separate images.
Rastering as data collection, yeah.
Right.
So
okay.
Translating and rotating at the same time.
You mentioned that the proteins in the crystals are multi-conformers. Can the R factor gap be reduced using the diffuse scattering information? At present, we only consider the Bragg reflections to refine our data.
Yes, I believe it can. The diffuse scatter is exactly what should make the difference between discriminating between what they call windshield wiper versus jumping jacks. So if you have two things in two different conformers, if they're doing this, is different than this if you're looking at diffuse scatter. So yeah, I think Nozomi has really been the first to demonstrate data quality good enough to start doing that. The problem with diffuse scatter is a lot of it comes from the lattice, which seems no one but me finds interesting. But yeah, the information should be there somewhere.
Well, that's terrific. And I have to congratulate you on perhaps one of the most stimulating talks I've ever heard in terms of how many different topics you hit and how much thought you've given to me. And I don't even do macromolecular crystallography.